You are at the cutting edge of cardiovascular research. I'm your host, Li Lai. I'm assistant professor of the Department of Cardiovascular Research. So here with me today in the studio is our colleague and assistant professor, Dr. Anahita Majuri, in our Department of Vascular Research in the Houston Methodist Research Institute. So Dr. Anahita Majuri, do you want to introduce our distinguished speaker today?
Sure. Thank you, Lily. It's my honor to introduce our guest, although I must say our distinguished guest today hardly needs an introduction. Dr. John Cooke trained in cardiovascular medicine at the Mayo Clinic, where he also earned a PhD in physiology. He was recruited to the Harvard Medical School as an assistant professor of medicine and later to Stanford University to develop a vascular medicine program. At Stanford, he served as a professor in the Division of Cardiovascular Medicine and associate director of the Stanford Cardiovascular Institute. In July 2013, Dr. Cooke joined the Houston Methodist Research Institute, where he holds several prominent roles, including professor and chair of the Department of Cardiovascular Sciences, director of the Center for Cardiovascular Regeneration, medical director of the Center for RNA Therapeutics, and chief translational science officer. His contributions to the field are truly remarkable and numerous, but with the limited time we have, we are excited to hear about his research and insights on cell-phase transitions in the cardiovascular response to ischemia. Welcome to our show, Dr. Cooke.
So thank you, Dr. Cooke. We're looking forward to hear more about the topic of my most interested topic, which is transformation, your talk. But before that, we want to hear a little bit more about your career path, so which I think it will be very helpful to guide our futures. So as Anna mentioned that you used to be a physician, and now you are a full-time researcher. So I'm curious about how you made this transition between full-time physician to full-time researcher, and how your experience as a physician affects your basic research.
Well, thanks, Dr. Lai and Dr. Majuri, for having me on this show, and it's a pleasure to be with you today. So I've always been interested in biology as a kid, and when I went to college, I was interested in neurobiology, so I majored in that. And then subsequently, I went to medical school in Detroit, my hometown, and from there, I went to the Mayo Clinic. At the time, I wanted to be the best doctor that I could be, and looked for the best training programs, and Mayo was at the top of the list, and I did get a spectacular training there. But about halfway through my training, I realized that there was something missing. And what was missing was an understanding of mechanisms. I really was interested in how things work. And although I love medicine and continue to love taking care of patients to this day, I really wanted to understand a little bit more about the reasons that we were doing what we were doing. And so I decided to spend some time in a research lab and loved it. I studied in the laboratory of Paul Van Houten and John Shepard, and there I learned about vascular physiology and pharmacology. From there, I went to Harvard, and as a junior faculty member there, I had my own laboratory and focused on vascular biology and vascular medicine as a clinician. So I tried to combine the two areas of interest, my interest in vascular biology, together with its application in vascular medicine. And pretty much I continued to do that for the rest of my career. I went to Stanford after that, after my faculty position at Harvard. I was recruited to Stanford and spent most of my career there until I got here to Houston to take over as chair of the Department of Cardiovascular Sciences.
Thank you very much, Dr. Cooke. The floor is yours.
Also, before we go ahead to your presentation, I think If the audience have any question that want to ask Dr. Cooke and interact with us, you can join us by two ways. First is you can join by web. You can go to polev.com and enter DeBakey, DEBAKEY, and respond to the activity. Or you can join by text. You can text DeBakey to 37607 and text in your message. So Dr. Cooke, looking forward to your talk.
Thank you. All right, well, let me share my screen. What I'd like to talk today to you about is something I find really fascinating, and this cell fate transitions in the cardiovascular response to ischemia. We're learning about how these cell fate transitions play a physiological role in the adaptation to reduced blood flow. And I'm going to introduce a couple of new concepts today. One is this concept of transflammation. And it's very interesting, and I think it has very broad physiological and pathophysiological manifestations, implications. And what I mean by this term, transflammation, is to describe the effect of inflammatory signaling on chromatin accessibility and cell fate transition. So when a cell is confronted by a challenge, basically this process allows the cell to reach into its genetic toolbox and pull out whatever it needs to survive or to adapt. And we know some of the elements of this pathway of transflammation. It begins with sensing the challenge, and pattern recognition receptors, like toll-like receptors on the cell membrane, can sense pathogens, for example. Pathogens present PAMPs, pathogen-associated molecular patterns that are sensed by the toll-like receptors. For example, bacterial polyliposaccharide, LPS, is sensed by toll-like receptor 4. So our cells can sense the challenge of a pathogen. They can also sense the challenge of damage. So oxidative stress, changes in pH, mechanical challenges, these can result in damage that represent damage-associated molecular patterns that can also be sensed by the cell. Example here in this diagram are the RAGE receptors, receptor for advanced glycosylation end products. Advanced glycosylation end products can be a signal for cell damage. In patients, most clinical representation of this is the diabetic with elevated glucose levels causing abnormal glycosylations of proteins. And that can be sensed by the cell as well. So anyway, these pattern recognition receptors, like the toll-like receptor, like RAGE, they, when activated, stimulate inflammatory pathways that are very well characterized. These inflammatory pathways are known to result in the generation of inflammatory cytokines and chemokines. We found another limb of the pathway working through this inflammatory pathway, NF-kappa B, IF3, other inflammatory transcriptional activators and coactivators are induced to cause changes in the epigenome, cause an increase in chromatin accessibility that allows for cell fate transition. So cells can change their phenotype in response to a challenge. Now, this process is very important, as I'll show you in just a moment, in the induction of pluripotency. So the ability to change a fibroblast into an induced pluripotent stem cell requires this transflammation process. Similarly, the transdifferentiation of a fibroblast to an endothelial cell also requires this transformation process. So let me show you some evidence to support that. But before I do, I want to introduce another term, and that is angiogenic transdifferentiation. This is a new process in angiogenesis that we've described that takes advantage of this transformation pathway that I just mentioned. Angiogenic transdifferentiation contributes to vascular recovery and describes a process by which a subset of fibroblasts, under the influence of inflammatory signaling that increases chromatin accessibility and the generation of angiogenic factors in the hypoxic milieu, can induce the fibroblast to become an endothelial cell. So the idea is that the inflammatory signaling increases DNA accessibility, and then the cell can respond to other factors in the environment, such as angiogenic factors, and that together, the increase in chromatin accessibility and the angiogenic factors can cause a change in the phenotype of the cell from a fibroblast to an endothelial cell. Now, another concept that I want to introduce is the idea that this inflammatory signaling works through multiple mechanisms to increase chromatin accessibility. And one of those mechanisms is a change in metabolism. So we've shown, and Li Lai, this is your work, has shown that innate immune activation can induce glycolysis. So fibroblasts under the influence of innate immune activation will increase glycolytic metabolism. And what we've shown is that the citrate generated in the glycolytic, in this glycolytic pathway is exported to the nucleus, where it is used by ACL to produce acetyl-CoA for histone acetylation. So that was some very nice work by Li Lai in the laboratory showing that this histone acetylation is critical for the cell fate transition. Well, the story begins quite a few years ago when I was still at Stanford. We were galvanized by the work of Shinya Yamanaka, who showed that fibroblasts can be induced to become iPSCs, induced pluripotent stem cells, under the influence of transcriptional factors. He used the four, OCT4, SOX2, KL4, c-MYC. There's other combinations that have been used. But he was the first to document that these four master regulators of pluripotency can, when overexpressed in a somatic cell like a fibroblast, can cause it to assume these features of pluripotency. The pluripotent cells that are made in this way can become any other cell in the body and can become endoderm, mesoderm, ectoderm, and that quite amazing discovery really galvanized the whole stem cell field, including our group. And we were interested to see if we could generate iPSCs using different methods. Shinya Yamanaka used a retroviral vector. We wanted to use non-integrating technologies, and that work led to an observation that was published in Cell. Ironically, the same month that Shinya Yamanaka got his Nobel Prize, we showed that he had the story half right. The master regulators OCT4, SOX2, KL4, c-MYC, those are absolutely critical for the generation of iPSCs. They're master regulators of pluripotency, and Shinya Yamanaka, of course, deserved the Nobel Prize for documenting that. And building on it, we showed that his retroviral vector was actually half the story, because what he did not know at the time was that the retroviral vector activated this transformation process, activated toll-like receptor 3 to increase chromatin accessibility so the Yamanaka factors could actually work. Without that increase in the inflammatory signaling and chromatin accessibility, you cannot generate iPSC. So that limb of the pathway is absolutely critical for this cell fate transition. Here's some of the data here we're generating iPSCs using the Yamanaka technique. So in this technique, you basically transfect the cells twice with the retroviral vector encoding OCT4, SOX2, KLA4, and c-MYC. And in panel B, you can see that you get these colonies 4 weeks later. If you expose fibroblasts to that retroviral vector encoding the 4 Yamanaka factors, you'll get these large colonies that contain hundreds of cells that are very different looking from fibroblasts. They are round, their nucleus is large, and they are pluripotent. When we blocked elements of the inflammatory signaling pathway, we didn't see any effect of MIghty88. That's an adapter for all of the toll-like receptors except toll-like receptor 3. When we knocked down toll-like receptor 3, which is activated by retroviral vectors, then we're not getting iPSCs. We knock down the adapter for TLR3 TRIF, and we also don't get iPSCs. So this was the first data indicating that the retroviral vector acting through toll-like receptor 3 is absolutely critical for the induction of pluripotency. There are other ways, non-integrating ways, to generate iPSCs, and we were among the first to show that messenger RNA encoding Yamanaka factors could, when delivered to fibroblasts, could also induce iPSCs. Now, this happens to be in murine embryonic fibroblasts. The colonies look a little bit different. These are colonies that you see in B at day 13, colonies of murine-induced pluripotent stem cells. So each of these colonies contain a few hundred cells, and they're pluripotent. And again, if we block MIghty88, we don't see any effect. But if we block the TLR3 pathway, either directly or by knocking down the adapter protein TRIF, we don't get we get a marked reduction in colonies. The ability to use mRNA, modified mRNA, for this purpose was really have to give credit to Katalin Karikó and Drew Weissman, who won the Nobel Prize for their work in 2023, showing that the modified nucleosides are absolutely critical in reducing innate immune activation so that the cells can actually tolerate the messenger RNA, and so that the messenger RNA is actually translated. Even so, even with the modified nucleosides, there is some activation of inflammatory signaling, and that is sufficient to activate this transflammation process to increase chromatin accessibility and permit the Yamanaka factors to act. Another way we looked at generating iPSCs is we embedded a doxycycline-inducible cassette encoding the Yamanaka factors in murine embryonic fibroblasts. So with the addition of doxycycline, you can see in the first panel on the left, you start to see these colonies popping up because doxycycline has activated that cassette. The cells are expressing the Yamanaka factors, and they start to turn into iPSC colonies you see here. Stained in red, this is an alkaline phosphatase stain. The iPSC colonies do express alkaline phosphatase. When we further activate TLR3 with poly-IC, which is a TLR3 agonist, we see more colonies, again showing the importance of that TLR3 pathway. If we knock down inflammatory activation with a decoy oligonucleotide against P65, we see fewer colonies, and we can basically abrogate colony formation if we knock out that inflammatory signaling pathway. So again, multiple ways we demonstrated that inflammatory signaling was absolutely critical for generation of iPSCs. Later, we showed that there's a Goldilocks zone for inflammatory signaling. Basically, the idea is that with too little inflammatory signaling, you don't get iPSCs. With too much inflammatory signaling, you don't get iPSCs. There's a zone of inflammatory activation that seems to be physiological and enhances chromatin accessibility for these fibroblasts when we're generating iPSCs. Here on the right side, again, are those doxycycline-inducible murine embryonic fibroblasts. They have that doxycycline-inducible cassette. We add poly-IC, we can get more colonies, up to a point when we add poly-IC over 300 nanograms per ml, we start to get fewer colonies. The cells are still alive. They're still responding to the poly-IC, and you can see that in the blue line. The blue line is showing luciferase activity. These doxycycline-inducible MFs also had a cassette encoding NF-kappa B driving luciferase. And with increasing poly-IC, we saw increasing luciferase activity. So we're getting increasing inflammatory stimulus, but up to a point, there's a reduction in colonies. On the other hand, if we block down inflammatory activation with the P65 decoy, on the left-hand side of this diagram, you can see fewer colonies and less NF-kappa B activation. And Palashanda showed that, in fact, that physically, that using a micrococcal nuclease assay, that there was an increase in DNA accessibility and a Goldilocks zone for that. So the basically, let me show you, have you looked at the right-hand corner here, that panel on the right. Basically, we have a little assay here to assess the DNA accessibility. A micrococcal nuclease will attack open chromatin regions and basically slice up the DNA at those open chromatin regions, and you get, fragments of the DNA. If the chromatin is quite open, you get a lot of mononucleosomes or dinucleosomes. And if it's less open, you get, polysomes. You get trinucleosomes and other polysomes, higher order polysomes. And so we basically here, what we did is we created a ratio of monosomes or mononucleosomes or dinucleosomes to trinucleosomes. So more open chromatin to less open chromatin. And you can see that the ratio is highest in this Goldilocks region that we just described in the previous slide. Well, this is all very nice with respect to understanding how iPSCs are generated. As a vascular biologist, I was interested to understand if this phenomenon played any role in vivo. So in the vasculature. So we started out first in vitro with see if we could change fibroblasts into endothelial cells with this knowledge that we had using an inflammatory activator, poly-IC, and then exposing the cells to factors that you might see in the setting of ischemia, like VEGF. And using that regimen and poly-IC, exposing the cells to inflammatory activation, and then exposing them to an angiogenic cytokine mixture cocktail, we were able to transdifferentiate a small percentage of fibroblasts to endothelial cells. The facts is sort of shown there in B. Not a lot of induced endothelial cells, to be sure, but they were true endothelial cells that we derived from those fibroblasts. And you can see that in the heat map of the transcriptional profiling that was done, comparing fibroblasts to the induced endothelial cells that were derived from those fibroblasts to genuine human microvascular endothelial cells. And you can see that the induced endothelial cells from the fibroblasts were much more similar to human microvascular endothelial cells by transcriptional profiling. And we could also demonstrate that functionally. So these induced endothelial cells made CD31. Fibroblasts don't. You can see that in panel D. Whereas human microvascular endothelial cells do make CD31, express CD31. You can see the immunohistochemistry there in E, and in F, uptake of acetylated LDL, that's an endothelial function. G, you can see the the networks in matrigel that endothelial cells form. And in panel H, you can see that these cells generate nitric oxide, just like human microvascular endothelial cells, whereas fibroblasts do not. That's in panel H. Now, another thing that is another factor that plays a role in inflammation is inducible NO synthase. Inducible NO synthase is activated by NF-kappa B at sites of inflammation. It's quite different from endothelial NO synthase, which produces small physiological amounts of nitric oxide to induce vasodilation. Inducible NO synthase has 1,000-fold greater catalytic activity than eNOS. It produces a lot of NO and a lot of superoxide anion because it runs out of arginine to donate electrons to. So you get basically the formation of peroxynitrite anion, which is a combination of nitric oxide and superoxide anion. And the iNOS is very good at nitrosylating proteins. So here you see under control conditions, fibroblasts that are expressing a polycomb complex protein, ring 1A, and inducible NO synthase. If you look closely at that slide, inducible NO synthase, the iNOS is all in the cytoplasm. You don't see in the nucleus, as you can see with the DAPI overlay. There's not really any iNOS in the nucleus. But with poly-IC treatment, first of all, you see a lot more iNOS being generated by these fibroblasts. You see it in the nucleus, and that's seen in the overlay. So iNOS is translocating to the nucleus. What is it doing there? Well, I can't go through the whole story. I don't have the time. But what we found was that iNOS translocates to the nucleus, and it actually nitrosylates epigenetic modifiers and plays a role in epigenetic plasticity. As shown below in panel A, you can see that poly-IC increases the generation of H3K4 trimethylation. That is an open chromatin mark. That increases chromatin accessibility. It reduces H3K27 trimethylation. That is a mark of closed chromatin. And with the iNOS knockout cells in panel B, now you see poly-IC doesn't have that effect to open up the chromatin anymore. H3K4 trimethylation is not increased. H3K27 trimethylation is not decreased. So the point of this paper was that iNOS plays a role in this epigenetic plasticity by translocating to the nucleus and nitrosylating proteins that are involved in chromatin accessibility. Well, does this phenomenon occur in vivo? And the answer is yes. Xu Meng was the first to show in my lab that fibroblasts can undergo a cell fate transition to endothelial cells and play a role in the recovery from ischemia in the murine hind limb model of peripheral arterial disease. So let's take a quick look at some of those data. This was an interesting experiment where she's taking collagen and embedding in the collagen human fibroblasts that are labeled with YFP. So you can put these plugs into immunodeficient mouse so that the human cells can survive. They're getting nutrition with the vasculature that's migrating in. And under the influence of poly-IC added to that nodule, you can see more vessels forming in panel A. With angiogenic cytokines, of course, you see more. With the combination of poly-IC and angiogenic cytokines, you see a lot more vascular influx, as shown by the blood that's in that plug. Well, most of the vasculature in these plugs that are in the mouse, most of them are murine vasculature. But we did observe that some of those human fibroblasts were being converted under the influence of poly-IC and the angiogenic cytokines. They were being converted to endothelial cells. So we were actually able to detect human endothelial cells in this plug where we had only inserted fibroblasts. And that's shown in panel D in the FACS analysis. So with the condition of poly-IC and angiogenic cytokines added to the plug, we saw more of those human endothelial cells converting to endothelial cells, human endothelial cells. Not a lot, but they were there. So that the phenomenon can occur in vivo. So there was evidence for that. But does it play a role in vascular recovery of ischemia? It does. Here's data using a different approach, again in the mouse, where we use the hind limb ischemia model of peripheral arterial disease in the mouse, where we tie off the femoral artery. These mice were lineage tracing mice where the fibroblasts were labeled with YFP. So at the end of the experiment, we can extract cells from the ischemic limb and look for fibroblasts using that YFP marker that have become endothelial cells just under the influence of ischemia. The idea being that you tie off the femoral artery, you're inducing severe ischemia to that limb, damage ensues, inflammation ensues in an ischemic region. And we thought, okay, here, those are the elements that could induce transflammation and cause fibroblasts to become endothelial cells. And they do. Fibroblasts become endothelial cells. Some subset of fibroblasts do. And you can see that in this very nice FACS analysis where you're looking at YFP, that's YFP positive cells are the fibroblasts. CD144 is a very good marker for endothelial cells. So in the right lower quadrant, you have YFP cells. Those are fibroblasts that are CD144 negative. So most of the fibroblasts, the YFP positive cells, are not expressing the CD144 marker. They're not endothelial cells. They're fibroblasts. But you can see that a significant amount in the top right corner, the right upper quadrant, of fibroblasts have become endothelial cells. And it's a significant amount because if you look at the left upper quadrant, you can see the true endothelial cells. So those are CD144 positive, YFP negative cells. So of all the endothelial cells that are in that ischemic limb, this study would suggest that about a third of the endothelial cells were derived from fibroblasts. So that's pretty impressive. And we have had confirmation of that by other investigators in the laboratory. Well, what subset of fibroblasts are playing a role in this angiogenic transdifferentiation? When we did single cell analysis of the fibroblasts in the hind line, we found that there's 8 subsets of fibroblasts in the hind limb by transcriptional profiling. 8 distinct clusters of fibroblasts. And what we found with ischemia was that subset 5 and 8 increased. That's shown in panel B. Subset 5 and 8 are increasing with ischemia. So we're interested in what those subsets are. We're able to use surface markers from the transcriptional profiling, surface markers to isolate those cells, FACS sort them from the ischemic hind limb, and then look at them in cell culture. And what we found was that the subset 5 at 28 days, that's the middle panel. The
Sorry,
middle
Dr.
panel
Cooke, if I may,
shows the cells.
if
Sure.
I may interrupt to ask question, it's amazing process. I have actually 2 questions. Does it, especially when you showed it in the mice model, do you think the age of mice would affect the efficiency of this process? That's one. And second, you showed the subset of fibroblasts that could contribute to this process and become induced, generate a new endothelial cells. What is new? What they have that the other fibroblasts doesn't have?
Two great questions. First question, does age influence this cell-fate transition? I think it does. We haven't really studied that very specifically, and we should. Certainly, as you get older, both mice and men, mouse and humans, there is an impairment in resiliency generally, and there is an impairment in angiogenesis. Wound healing, for example, in older folks is impaired, and that is in part due to an impairment in angiogenesis. So I do think that this ability, this resiliency in response to a challenge, this ability to undergo this cell-fate transition requires a plasticity that may be adversely affected by aging. It's a really great question. It's something we should study. That's a nice R01 right there. The effects of age on cell-fate plasticity or cellular plasticity. That would be a very cool one to look specifically at this, how this phenomenon of transformation is affected by aging. Your other question was, what is different about these clusters? So cluster 5 and cluster 8 are different from the other fibroblasts in that their numbers increase with ischemia. So that was one clue that they're different. And they have a different transcriptional profile. The single cell sequencing suggested that cluster 8 made angiogenic cytokines. Cluster 5 had some elements of endothelial identity. There were some surface markers, for example, that we normally associate with endothelial cells that were expressed by the C5 cluster. And then when we isolated these cells and studied them in two-dimensional culture, they all look like fibroblasts. And I'm sorry, I apologize. These are not the best microphotographs, but I think you can see in the very first panel, upper left-hand corner, these are fibroblasts from a non-ischemic limb. 0 days means the limb was not ischemic. 28 days is 28 days after the femoral artery ligation. But at 0 days, in a control limb, you can isolate these fibroblasts that look like fibroblasts. And then if you look at them in matrigel, we're looking at the upper row, the third figure, the third panel, upper row. Those 0-day fibroblasts, those are just general fibroblasts isolated from that hind limb that's normal. They form nodules in matrigel, and that's typical of fibroblasts. They form nodules. Now, if you look at the go all the way to the bottom, and you look at the 28-day cluster 8 fibroblasts. They also form nodules too. If you go to that again, that third panel in the bottom row, they look like fibroblasts. The fibroblast response to matrigel is to form these nodules. Now look in the middle panel. In 2D culture, the 28-day cluster 5 fibroblasts look like fibroblasts in 2D culture, but when you put them into matrigel, they start forming these networks that look very similar to the networks we see with endothelial cells. So endothelial cells form these networks in matrigel. So to answer your question, the cluster 5 and cluster 8 cells are different. Cluster 5 express some endothelial markers, and then in matrigel they form these networks. Cluster 8 seems to produce angiogenic cytokines. Well, again, I have to give credit to Dr. Lee Lai for introducing this concept of metabolic control of cell-fate transition, where she showed that the inflammatory signaling, in addition to everything else I've shown you, inflammatory signaling increases glycolysis, and the increase in citric acid, citrate, in the mitochondria during glycolysis is transported out into the nucleus of the cell, and is converted to acetyl-CoA to participate in histone acetylation. Histone acetylation is a critical process in open chromatin. So sites of open chromatin have more of that histone acetylation. And she went on to show very nicely that if you block the conversion of citrate to acetyl-CoA, you get an impairment in the recovery from ischemia. So again, this is our femoral artery ligation model. We can measure blood flow with laser Doppler. And in panel B, you see the severe drop at day 0 after the femoral artery ligation in both groups. But there's a recovery in both groups. It's blunted in those animals that receive the Bristol-Myers Squibb compound to inhibit the conversion of citric acid to acetyl-CoA. You can also see that in panel C in the assessment of capillary density, nice capillary density in the skeletal muscle of the hind limb, shown in the left-hand side in the vehicle-treated group. On the right-hand side, the BMS-treated group, the capillary density is reduced. So that suggests that this process of metabolic regulation, this process of transformation is a metabolic regulation. Shuang Li has gone further to knock out the enzyme responsible for converting citrate to acetyl-CoA. That enzyme is ACL. And so in her fibroblast-specific knockout of ACL that's shown in panel A, how she did that, confirmed in panel B, showing the knockdown in ACL with tamoxifen. In panel D, you see the effect of that with a blunting of the vascular recovery in the ACL knockout, fibroblast-specific knockout. And I'm going to leave this slide for you to talk about, Dr. Lai, in the discussion. A new concept in metabolic control of cell fate transition. So I'd like to end by just again describing these two terms. Angiogenic transdifferentiation is a new process in angiogenesis that we described that is due to a combination of inflammatory signaling that increases DNA accessibility and a glycolytic shift that increases histone acetylation. The inflammatory signaling also increases NF-kappa B translocation to the nucleus to open up the chromatin. In any event, this inflammatory signaling, the metabolic regulation, participate in generating induced endothelial cells in the setting of ischemia. And those induced endothelial cells contribute, from fibroblasts, contribute to microvascular structure perfusion recovery, vascular recovery from ischemia. And finally, this concept of transformation, that cell transitions can occur in response to a challenge. So basically, a challenge can be sensed by the cell, either through pattern, through pathogen-associated molecular patterns or damage-associated molecular patterns in the case of ischemia, to activate pattern recognition receptors like the toll-like receptors, like the RAGE receptors, inducing inflammatory signaling that causes changes in expression of epigenetic modifiers. We didn't talk about that, but there's an increase in histone acetyltransferases. There's a reduction in histone deacetylases. There's alterations in the activity of polycomb complex, and there's a metabolic coupling that occurs with that, all of which contribute to an increase in DNA accessibility. And that allows the cell to reach back into its genetic toolbox and pull out whatever it needs to survive and adapt. And sometimes that requires fluidity of cell phenotype and a cell fate transition. So I have a remarkable group of young scientists that are working with me, and great administrative group and faculty, and well-funded by NIH and non-government organizations, and great collaborators that have made all this possible. So thank you, Dr. Lai and Mojiri and the Methodist studio there for giving me a chance to talk about this work.
Thank you very much, Dr. Cooke, for the really insightful, thought-provoking presentation. So I guess Dr. Le has several questions.
Yes, thank you so much for the talk. So in the first part of your talk, you introduced the Goldilocks zone of the inflammatory signaling, which I think is very important. I think the finding we have will certainly instruct us and clinically how to use this anti-inflammatory drugs. So what do you think about it?
Yeah, no, I think that's a great observation. I do think clinicians have been aware of this Goldilocks zone for a long time. Maybe not cognizant of the mechanisms, but aware that in the setting of excessive inflammation, like a diabetic foot ulcer, where you have a ring of inflammation around the ulcer, healing doesn't occur. If you remove that ring of inflammation with debridement, you can sometimes observe an improvement in the healing. Also, on the flip side, when you have an insufficient inflammatory signaling, such as in patients that are on steroids, healing also is poor. So a certain amount of inflammation is necessary for healing to occur, for rejuvenation, regeneration. You need a certain amount of inflammatory signaling to give the tissue the regenerative ability, the fluidity of cell phenotype to allow the regeneration to be completed.
So to follow up with this question, so you used a highly ischemia model to identify this novel transdifferentiation phenomenon. So do you expect or you already see an impairment of transdifferentiation in the diabetic mice? I think diabetic mice has too much inflammation.
Well, to be sure, it's well known that diabetic mice have an impairment in healing that mimics the impairment in healing of diabetic patients. I think it would be interesting to look to see if this process of transformation is impaired, if the ability to undergo cell-fate transition is impaired. That would be a worthy project. And it could also lead to some insights into pathophysiology of diabetic foot ulcers that might be useful. Might have give us insights into a new therapeutic avenue.
I was wondering, how do you think you can enhance this transdifferentiation to have more endothelial cells that can enhance the repair and regeneration, enhance the wound healing? What would you think? Either enhancing, because you showed several papers that it could be regulated by metabolism, it could be regulated by the methylation of the RNA, it could be regulated by different factors, and it has a very specific window. How do you think you can enhance this process in a way that you have more endothelial cells to incorporate to the area of injury and enhance revascularization?
Yeah, that's a great question. I think we have filed some intellectual property on agents that can enhance the histone acetylation through these metabolic pathways. I do think that the pathways that we've uncovered could lead to new therapeutic approaches for the treatment of peripheral arterial disease, for healing wounds, almost for any regenerative phenomenon. I think that this process of transformation is critical, and I think understanding these mechanisms can lead to new therapeutic avenues.
I think my question is also related to Anna's question. So, since in your circulation paper you found that there's a very small percentage of endothelial cells that can derive from fibroblast cells. So, what is the reason? Is it due to fibroblast heterogeneity? And if so, do you think this, if this fibroblast cells, those population of fibroblast cells that can turn into endothelial cells, are they predetermined or they are inducible, or if this population is a dynamic population?
Yeah, so great questions, and I do believe that. Well, I think the evidence does support from our group and other groups that there's a heterogeneity of fibroblasts, and there does seem to be a subset of fibroblasts that can more easily transdifferentiate into endothelial cells. And it's possible that fibroblasts can transdifferentiate into other cells as well to support regeneration of a tissue. I do think it's also dynamic. You asked that question. Are these fibroblasts phenotypes fixed in stone? I don't think so. I think it's these clusters that we're seeing. For example, in the mouse limb, we saw 8 different clusters of fibroblasts. I suspect that that is very dynamic. That fibroblasts can move from one cluster to another. And that dynamism might be very important for resiliency of the organism, and that dynamic nature of the fibroblast subtypes. I mean, that could also be impaired with disease, with diabetes, with aging. So good questions. I don't have the answer at the moment, but I think that would be something worthy of investigation.
In early of your talk, you mentioned that how important to have a very limited inflammation to have an open chromatin for cells to respond and transdifferentiate. So you're familiar with the inflammaging, which is aging associated with the inflammation. It is well described that as we age, the level of inflammation increased. So it is could you please elaborate on that we have quite a level of inflammation by aging, but all the process that you just described is a very it's not at all, if I say, efficient. So the wound healing and the process of regeneration is very slow.
That's an astute question, and I think what you're getting at is that, hey, look, in senescence and with aging, you get this phenomenon of inflammaging, which is the production of inflammatory cytokines by senescent cells. And so how is it then that a senescent tissue doesn't have the resiliency? Because what you've been saying is that inflammatory activation is critical for resiliency. It's critical for the ability of a cell to change its phenotype as needed with an injury or with a challenge. And my answer to that is the Goldilocks zone. Again, I think that explains the apparent paradox. I think it would be really interesting to look at this phenomenon that we uncovered, the mechanisms whereby excessive inflammation impairs cell fate transition. What's the mechanism of that? That actually understanding what it is that excessive inflammation does to cell fate transition, I think would be really a great avenue to explore, because maybe we can intervene there. Maybe we can reduce inflammation or reduce some limbs of the pathway, antagonize whatever it is that's impairing cell fate plasticity with excessive inflammation. So something about excessive inflammation is reducing plasticity, reducing resiliency. And I'd love to know what that is.
Thank you.
So I guess my last scientific question is, since you introduced this fundamental new phenomenon of transformation and transdifferentiation, so do you think in the future the strategy for us is to induce endothelial cells in vitro, or we try to do everything and enhance this process in vivo?
Okay, so that's a great question. And I guess what you're getting at is, should we generate therapeutic cells ex vivo and use those for therapeutic purposes? Or should we try to do this all in vivo, manipulate the cells in vivo so that they undergo the therapeutic transition that we would like them to undergo? Well, it is easier to modify cells ex vivo and then rather than in vivo. Modifying the cells in vivo is maybe more difficult. On the other hand, delivery is a problem for therapeutic cells, always has been. We don't know what the right dose and duration, timing, dosing. The other problem is that you put cells into an ischemic environment. That's a hostile environment, and cells don't do well in that hostile environment. I think it's a better strategy to try to figure out how to modify the cells in vivo, modify the autologous cells that are in the right environment, in the right place, rather than trying to inject cells. I think it's just more difficult to try to use cell therapy ex vivo. I think probably better approach to figure out how to manipulate cells in situ.
So thank you, Dr. Cooke, for all the insightful talk scientifically. So I think, do you have some last wisdom to people who, like me and Anna, who just start off in our independent career? So what's the most important quality you think for us to cultivate to be able to survive in this competitive world of getting grant, getting successful?
Well, there's a lot of skill sets that you need to be successful. Persistence is an important one. Grit, the ability to overcome challenges. Being open to ideas. Collaboration is really important. Being able to collaborate with people. Being fearless. Take on new technology. Be open to new ideas. And get the right people to work with you. That's really critical. I think I've been lucky over the years because I've had great people to work with in great environments, and that plays a big role in how successful you can be. It's other people, really.
Thank you, Dr. Cooke, for your talk and also your suggestions. So we're really grateful to have you in our show.
Yes, thank you very
Dr.
much.
Cooke.
My pleasure. Thank